we have a defective gene, we engineer the corrective gene and take a virus, strip out all its harmful characteristics, put this correct gene into that virus, which is now just a vehicle that's carrying the corrective gene, inject it into the patient.
Today we had Poorvaja Jagadishan.
And now I'm working as a manufacturing technician at the GTIMC, which is basically the Gene Therapy Innovation and Manufacturing Center. It's a spin-out company from the University of Sheffield. They mark a batch manufacturing record that thick. So then note down everything. There's once you finish your process with the team, you have to clean the entire place. Not normally cleaning. Nope.
Poorvaja has gone real deep into what goes on in a day in her work.
Exactly. It's a typical startup environment. I am so blessed to have started my career here.
True.
So it's like, this is the first time I'm working, but I'm working in this environment. Is it supposed to be this? Okay, probably it is. And I got a rejection, which was 3 pages long. Very new. So I read through it. All it said was, we loved you. We feel like your skills will be more suitable towards a manufacturing technician role. And we will be advertising for that very soon. So please do apply.
Okay.
12 months experience in the industry, amazing. Really good. If you have that, if you can have that, well and good. Companies initially, I started applying since June. And when I applied, basically, I think it was rejection.
Your video.
And in your cover letter, all the recruiter is looking for is how you say these skills could be transferred to this job that I'm doing. Explain that in your cover letter.
Hello, welcome to Analytics and welcome back to another episode of the podcast series. Today we had Poorvaja Jagadishan, who's a manufacturing technician in Gene Therapy and Innovation Center, which is affiliated with the University of Sheffield. If you are somebody who's looking for jobs, you know how it is important to know what is in a job description and what does a job entail to. If you're interested in gene therapy, this is a video you need to see, because Poorvaja has gone real deep into what goes on in a day in her work. Enjoy your video. Poorvaja, welcome to Analytics.
Hello.
Hello. Long time no see.
Yeah, long time.
Good to meet up with you though.
Good to meet you too.
Yeah, thank you. So we'll start off with an introduction. You can introduce yourself, what you're doing, where you're working right now.
Okay. So I'm Poorvaja Jagadishan, and I moved to the UK like 2 years ago to do my master's in advanced cell and gene therapy at the University of Sheffield. And now I'm working as a manufacturing technician at the GTIMC, which is basically the Gene Therapy Innovation and Manufacturing Center. It's a spin-out company from the University of Sheffield. We produce viral vectors that carry the gene therapeutics for patients with severe genetic disorders. So we mostly produce clinical grade vectors that go into preclinical and clinical trials.
Right, vectors in the sense?
Viral vectors. So gene therapy is basically any genetic disorder is a defect in a specific gene. So what we do in gene therapy, in very simple terms, is we have a defective gene, we engineer the corrective gene and take a virus, strip out all its harmful characteristics, put this correct gene into that virus, which is now just a vehicle that's carrying the corrective gene, inject it into the patient. So now that is a replacement for the defective gene. And so it's going to produce the right proteins and eventually you cure the patient, but not just treating the symptoms, but the root cause is gone now.
Right, so it's like a custom-made solutions for each patient?
It's not for each patient. So you can engineer it, like extremely custom-made. I think that's called precision medicine or something. People do it. That's kind of not what we do. For us, it's like a big disease. So, for example, sickle cell anemia.
Right.
It covers a huge population. They're all going to have the same genetic defect. So if we just find the target, and that's the same thing that we're doing, we're going to produce one gene, correct gene, and that goes into each and every patient.
Right. So it's like, is it like a part of the gene composition that's affected by a disease? That might be similar in a viral audience,
Yeah.
wider people.
Exactly.
That's what you're.
Yeah, we target that. Yeah, as I said, let's take the example of sickle cell anemia. It's probably this will help. So it is basically, I'm not sure if people without science
No,
background
it's
will
all right.
understand this, but we have several amino acids in our body, so they kind of make up each and every gene. So what happens here is a single amino acid replacement that causes sickle cell anemia, basically. Right. So we are just producing the right gene to alter that.
Right.
That's
right.
it. So we just have to figure out what the common target is. Finding a common target and then making a gene for it, the corrective gene, putting it into the patient, and that works. That's
Right.
it.
And how far long has it been used for?
So this was actually found years ago. I think in 1900s, it was introduced, and it was actually first introduced in the US. The UK had used it. Even China had some gene therapies. But somewhere between, I think mid 19th. I'm not sure if I'm getting the years correct because I read this a long time ago. But somewhere in mid 1900s. Again, as I said, this is like you are fighting a target, you're giving it to the patient, the same doubt that you had. What if it goes wrong?
Right.
So things could go wrong, and there was something that went wrong. I think that it was for a cancer, a type of cancer. Though the gene therapy cured the cancer, it kind of had an off-target effect. It went and targeted some other gene also. So I think 10 boys, they were affected. They got some other cancer.
Okay.
So that was a huge setback, and everything was shut down.
Right.
And then now we've started it again, like trying, researchers are doing like a lot of researches to understand what's going on with this virus. Is it clipping the correct target?
Right.
The main thing is it has to go to the target, not just move to other organs. So because of the advancements that we have now, we kind of know more about it.
Right.
So that's why people are starting back, and gene therapy is kind of growing a lot, I'll say. I think
I mean, the whole med tech or med engineering is a big thing. Similar to AI and most of it's a bit more niche and not easily accessible by everybody. So, and I've seen a lot of people who belong to this section. That's why you're a good guest today. Do you want to tell me about your background? Specifically concentrating on what led to getting this job.
Okay.
If anything contributed from your history.
Yeah, okay. So I basically did my undergraduate degree in industrial biotechnology from Anna University. So that degree, it had a lot of modules, which was really stressful back then. It had engineering mechanics, engineering graphics,
Right.
thermodynamics, fluid mechanics.
That's like the core of mechanical engineering.
Yeah, I did all that.
Okay.
And I also did molecular biology, genetics. So it basically covered everything that is there. Because at that point, when I thought about it, when I was doing the degree for 4 years, I was thinking, what am I even doing here? Why am I studying like code mechanical engineering stuff here?
I mean, I wouldn't doubt that. Probably the core mechanical. I mean, I have a mechanical background. I did my bachelor's in mechanical engineering. So the core mechanical concepts and principles are really close to nature, and it's like the defining nature itself, quantifying nature itself. So it could be used anywhere, not just biomed, but go into any principle it could be used. So it's really good to know. I mean, I don't blame that system actually. To be honest,
at that point, I just had too many modules, and I was like, oh, fine.
Wait, are you finding any of those useful now?
Yes.
Anything? Right.
Yes.
You are, right?
Yes, I am. So that's what I'm trying. That's why I brought this up. So my undergrad was a lot of modules put together. And at that point, though it sounded irrelevant, it was not. So that was kind of the foundation, because I knew a lot of things. I can understand a lot of things because of that. So that's the first foundation. And from there, I have always wanted to pursue genetics. Like I wanted to become a genetic researcher. So for my master's, I was looking at genetic degrees. And even in my undergrad, all the elective subjects, I mostly opted for genetic engineering, all that background.
I like that. targeted
Yeah.
approach is what would kind of make you reach where you want to be.
Exactly.
I have been like spread out. I've been touching different things, but I'm kind of doing that now, targeting things and going into what I really want to do. Right?
Okay.
So it's working out for me as well. Good point that you brought up. Yeah. So after bachelor's,
So after bachelor's,
sorry, did you have any projects in your bachelor's? I saw some
Yeah.
papers that you published.
Yeah, so I did. I have some projects.
Yes.
So during my bachelor's, it was during COVID, point one,
Right.
and though we had a lot of hands-on lab-based experience in our degree, COVID kind of took a lot of the things that we could do. I was supposed to do an internship. I couldn't do it. So during that time, I was trying to just be more productive. And so me and my friend, we were trying to do review paper. It was not a research
Right,
paper,
yeah.
just a review paper. And like me, he was also interested in genetics, but he was more interested towards behavioral genetics and forensic genetics.
Right.
So we researched a lot, and we did a paper. I don't even remember the name.
The
of
criminal
the paper.
and psychology.
Yeah, it was related to forensic science. So it was basically researching what are the genes that could influence aggressive behavior
Right.
in people. So we wanted to take it far ahead. We wanted to actually go into prisons and actually interview people.
That's primary data.
We wanted to do that. We want to like interview people or focus on one big case, see if that
right.
person has some genetic background. So because in our heads, we are science-based people. So in our heads, if you have a standard aggressive nature, there is something inbuilt in you.
Right.
You have a gene. And I think it was nature versus nurture, the paper's name. It was nature versus nurture and something else. So it is your nature to do this, but can you be nurtured in such a way that your aggression could be brought down?
Right.
So at that point, I was looking at genetic counseling as my career. So because
Genetic counseling, what's that?
So genetic counseling is basically as I said, a lot of people have a lot of genetic issues. So if somebody is identified with a genetic disorder, the genetic counselor helps them to cope up with that. So it may be a normal amino acid deficiency disease, or it could be sickle cell anemia, it could be ALS, MND, motor neuron diseases. There's a lot of diseases. So, and most of them have a genetic basis.
Right. Motor neuron diseases, I think Citran here is
that was my project.
All right. Yeah.
That was my project there.
Yeah, go on.
So that was one other thing that helped me with my job actually. So so we did that paper. So the key concept was, yes, if you have an aggressive gene.
Really?
Yeah.
All right. So is that what the literature is saying overall as a whole?
The literature is not saying that. The literature is saying these are the genes that could cause your aggressive behavior. We have found studies that say, I think there's MAOA gene. Okay, there's one gene like that, and then even men are considered more aggressive than women in some cases. But that is again another thing. That is because there's a specific protein that men have, and that kind of increases your dopamine levels.
Right.
So you have higher dopamine levels than women. So that paper we kind of linked a lot of things in that paper, and even a little protein that in the Y chromosome, it can cause that increased aggressiveness in men. So we kind of linked all that in. So it was just like fun. Two kids who love genetics, having fun researching about genes, linking everything and anything. If you look at that, I think I did this flow chart in that paper. I don't remember out of the top of my mind like what it actually was, but I had linked like 3-4 genes that made no sense at all at that point. So there was like the SRSH gene, which men have, and then there was the MAOA gene, there was something related to serotonin. So it's just all these are basic genes that everybody has, and just an increase in level of this gene could result in an increased aggressive behavior. That's what we found.
Right. For disclosure, that's from the papers reviewed, and not based on primary data.
No, it was just a review paper. So that was the thing. It was two people having fun reviewing stuff. So it was based on data that was already expressed in literature. So we just took that and we kind of made
Right.
something out of it. That's it. But
That was your bachelor's.
yeah, that was my bachelor's.
And did your college or university promote these, or was there a culture of doing this in your university? Or
I think we were the
did
first.
you get?
I think
Yeah,
we
you
were
went
kind
out of the way.
Yeah, we kind of went out of the way. We were bored. We wanted to do something. We had a professor who actually helped us a lot. She was a genetics professor. She kind of liked us, so she was like, "Go for it, come on." So we did that, and that kind of helped us even more. I think my professor, she had something going on in her lab. They were doing a research using bromelain, which is a protein from pineapple, basically.
Okay.
So they found preliminary data that bromelain could help cure COVID. So, I mean, not cure probably, it could be preventative, it could help you, but there was data. So they wanted me to write a review paper on that now, because it was not my project, so it still is a review. So we did that then. That's the other review paper that I wrote. And then there was my final year project, which was something that we wanted to do on our own. It was a basic project, a very basic project, but we can call it our own. That was like, because it was not something that a professor is already doing.
Right.
So I think that project was saying these proteins from fruit can help to extract DNA. And this could be used in forensic science, because they are using a very costly proteinase K.
That's useful.
Yeah. So if we can just get it out of simple fruits that we have in the market, like I think we use kiwi, pineapple. There's already research for kiwi and pineapple. They've proved it. So our thing was, I don't know if not many people might know this, but you might know. Nartanga. I think it's called citron.
Right.
Okay. So it's kind of a citrus fruit. They use it in pickles down
Right.
south.
Okay. Yeah.
I don't know what they call it in Malayalam. I'm sorry. So Nartanga in Tamil, it's called
Is it a
Nartanga.
big one?
Yeah, it's like that.
Oh, it's like.
Green.
Oh yeah.
And they use it for pickle.
It has different names even inside Kerala. So,
Amazing.
oh.
There.
Yeah. That's good. Yeah.
So Nartanga, that we used that, and that kind of gave us really good results.
Right.
So that was kind of interesting. So that was another project. It was very simple for a 4-year industrial biotechnology degree, but it was something our own.
First off, your choice of degree was really niche.
Yeah.
And then you went out of the way to do other stuff, and that's something common I see in a lot of successful people. That's what you need, actually take initiative and do stuff. Right. And then how did you do something after that?
No, that's it. That's the end. So that's my undergrad. And then for my master's, as I told you, I wanted to do something in genetics. I had already applied and got my degree from my course from Edinburgh. I even paid the deposit there.
Right.
So after that, a random lecture from my 7th semester animal biotechnology professor. So it had like one paragraph about gene therapy. So I just got interested and I was like, what is this? Because I like new things. And that was the whole point of me wanting to go into genetic counseling. But it had a lot of extra things that I should do, and I didn't have the time to do that. So when I looked at gene therapy, I was like, okay, what is this? This is new. And I researched, and that is when the company that I work in right now opened.
Oh,
so that's how I came to know about this course. So it came through my company. So they had just opened the GTIMC, and I got curious, what is this? And then I checked out that the University of Sheffield offers a master's degree. Just applied for it and got the offer.
Right.
I think even the application process was kind of different. They actually asked me for like, I don't know what they call, these architecture students take portfolio. They asked me for a portfolio. Yeah. And I'm just there like, what is a portfolio? I remember literally going to my dad and being like, Dad, what's a portfolio? Like, how do I sell myself? I don't know, because I've never heard this word before. What do I do? So it was basically all my research projects and everything. But yeah, so that's how I came to know about this course. Got in, ditched Edinburgh, sorry, but came to the Uni of Sheffield, and that's how I got.
I think it's a good place to be if you're doing research. Specifically, if you're doing research and if you're getting into research, it's a really good place to be. They really mold you and really train you to do research. And they really focus on critical thinking, questioning things, not take it for face value.
Yeah, that's true.
That's one thing I really love about the university. Yeah, so you didn't walk in between your bachelor's and master's straight
No
away.
pressure.
Yeah, all right.
So you will hear a lot about those struggles also.
Yeah.
So I got in my master's. Did my master's. It was a 1 year degree. But I should say it. I think I was the second batch of doing this course. So it was an extremely niche course, extremely new. I was the second batch. There was only 9 people in my class, and the first batch had only 3 people. So you can imagine
Right.
how new it was.
All this in CTRAN.
Yeah, instant.
But I think CTRAN has got a good leader. She has like good insights on where she should take CTRAN. And CTRAN is like, even though people might not know University of Sheffield, they might know CTRAN.
Yeah.
That's how.
Citron does a lot of research, especially in M&D.
Yeah.
All their professors, to be honest, that's what I was about to say. Like, I know a lot of professors at Citron, and I can actually count how many preclinical trials are going on.
Right.
And I can say there's at least 4 preclinical trials that I know out of the top of my head that will go into actual clinical grade.
Yeah, I've heard a lot. I've had friends from something with motor neurons. They were doing research on that, so I've heard that as well. So that's why I thought you probably have a good leader who has good insights on where she should take it to. All right, I'll let you continue.
So the master's course was fairly new, but they did a really good job. We had a lot of professors talk about it, and the input that they had on the first day was, by the end of this course, you'll probably know more about gene therapy than us. Because we are doing research in one specific disease, but you are learning about all that. So by the end, you will know it all.
Right.
So that kind of a space, they are able to tell that you will know it all by the end. That kind of a space was something new for me. And mostly it was professors. They come in, talk about what preclinical trial they're doing, what is gene therapy, all that. And we also had a lot of people from industries coming in, talking about what are their projects, how is the world going on about gene therapy and everything, the regulatory aspects of it. So we had like a really good set of lectures and everything. And apart from that, the assessment pattern was something that I loved. And I think that, like, the money that I gave is purely for that. Because I wrote one exam. That's it. Just one exam that covers what is the basis of gene therapy. But then everything else was a poster presentation, a presentation.
Right, yeah.
But the thing is, the topic of the presentation was, you have to figure out a gene therapy for a disease that is not published anywhere.
Right.
You have to believe that you are a principal investigator, figure out the logic, figure out a target, how you're going to treat it, talk about the regulatory aspects, and it just increase step by step. First thing was just figure out a disease and a target that's not published. The next stage was, that's one presentation done, graded. Next stage, figure it out, but at the same time, you have to also think about regulatory aspects.
Right.
Then it was from bench to the market, how you're going to take it.
Right. It's like the whole life cycle.
Exactly, and you can't like repeat the same disease. So we have to think about different diseases, different targets, a completely new thing that's not
For
published.
each assessment.
Yeah.
That's cool. That's like a real world experience that you need.
Exactly. So I think that helped me the most, because that is the most my brain has ever thought. So that was a lot, because, and again, the panel is like 3 principal investigators who have been in the industry, who have talked with the MHRA.
So it's not even the professors who's going to assess this.
No, they are professors.
All right. I mean, here it's like that.
Yeah, so Citran is like, everybody's a principal investigator.
Yeah, right.
Everybody has their own clinical trial going on.
Right.
So all of them are sitting there, and they question you left and right. They're like, okay, how do you think this can happen? Like, this can happen. I'm going to stop my funding. What do you think? They are giving me all kinds of scenarios. It may be science, it may be regulations, it may be funding, and you have to answer there. So that kind of helped me a lot. So that was one of the best aspects of the course, and that kind of helped me to think more, think from a different perspective, and at the same time be confident in what I say. You're being badgered by 4 different people there every time. So yeah, that was a really good experience. And then the project that I did was, again, it was again a gene therapy. It was a preclinical trial for ALS, amyotrophic lateral sclerosis.
Was that because my other friends, they kind of formed part of an existing clinical trial.
Okay.
Was that like that, or was it like
This was This was it was a preclinical trial that my professor had
Had
started.
been doing, yeah. That's
He
very
started it. It was almost in the animal stage,
Mhm.
and it will go into actual clinical trials, I'm pretty sure. But yeah, I was really blessed to get into that. So it was by Dr. Guillaume Hodberg, or someone. So he kind of paved my career for me.
Right.
So that also helped me a lot because that was a preclinical study, almost in its animal stage. So they've done all the prior studies. I have data. All I have to do is learn it, learn how to do the experiments, and I worked with AAVs. So that's the viral vectors basically. So that is what we will be producing in my company.
Right.
So I kind of worked with that. I had the experience, and that project was really good. It taught me how things work in the UK, how research is done here.
Right.
That's kind of new. So all that, and also the writing bit, like thesis writing, how is that different and everything. So that was
I
good.
know that. Know that quite well.
Exactly. So yeah, it taught me all that.
Right.
And so after that was job basically. So all this, I think, foundation was my undergrad.
Yeah.
And then the confidence and some of the skills was
Yeah.
all the projects that I did. Masters, foundation for gene therapy,
Yeah.
probably. And the project that I did was the final nail, I'll say.
Nail in the coffin.
Yeah, it was perfect. And that's landed me to the job, basically.
Right. So, did you have any course mates who were from a different background other than? I mean, even you had a bit of a background
Yeah.
there. So, was there anybody who was like completely new to it?
Nobody was new to genetics, like genes
It was a
specific.
good process that they had selection for.
Yeah,
For the master.
but I mean, I think they still believe that people with no gene therapy or genetics background at all will come into the course because the first few classes that we had was the basics. Like, even if you go, you will understand because they were trying to.
I'm all in.
Because they were trying to tell us the lecture was what is a gene. We learned that in 12th standard.
Right.
Being a biology student, you learn it in 12th. So they were trying to like level the platform for everybody.
Yeah.
So it doesn't matter what your background is. If you want to do gene therapy and if you get into the course, you'll be fine.
Right. So and yeah. So what did they look in? Like if somebody from outside is coming, is it their drive to do research?
I think so. I think Citran mostly they want people who can do research. They want passionate people.
Right.
That's one thing I feel, especially in this field. If you're passionate, that gives you a lot of strength.
Right.
So they want you to be passionate towards it. Prior research experience could help. Even if it's academia, it's fine.
Right.
And then probably a little biology. Probably they might look at it,
Right.
but I'm not sure. But this was what they told us.
Yeah.
We want to level the ground. We don't want to assume that each one of you here know what a gene is. So
Right.
we will do that.
Yeah. Good. Cool. So that's done. Master's done.
Yeah.
And so how was the whole experience? Stressful?
Masters?
Yeah. Enjoyable?
It was not stressful. Like, I mean, I was probably stressed the day before the presentation or something, but it wasn't as stressful.
Right. Yeah.
Like, I'm used to a lot more pressure in terms of academics.
I think that's the one good thing we got from India.
Yeah, that's
we can
the thing.
survive things. I felt really easy.
Yeah, true.
The masters here, to be honest, wasn't stressful at all. to be really fair, I mean, if you understand what needs to be done, if you understand how we have to look at things, how you have to question things, just understand the basics and just remember that you're not giving an exam, but like criticizing things and think for yourself. Then it's quite easy, to be honest, compared to back in India. It's for my course. Might differ for others. So let's say if somebody is doing something in AI, maybe a bit more technical. If they're coming from a completely non-technical background, they might have to sit and learn the coding rules, and that's quite required. So that might take some time. Otherwise, compared to back in India,
I'll say that. So
Yeah.
the course was not so rigorous.
yeah,
I had days off.
yeah,
I probably like went to class 2 days a week, 3 days a week. That's also been there.
right.
I could have taken more pressure, and I can't blame them because in
Right.
India we are put through a lot.
Yeah, a lot. And this is more useful as well. You're doing stuff that you will do
Yeah.
in your job. My question is why is it not happening in India? I mean, do we not have enough resources? Do we not have enough researchers who come in as professors? What's going on? What do you think?
What do you mean, like, not happening in India? What aspect of it is not happening
I
in
mean,
India?
why is it? I mean, do you think as institutes like this and researchers like this happens in India on a regular basis?
No.
No,
So from personal experience, I can say
yeah,
that.
personal
So
experience. Just personal experience. You're not aware of the whole thing going on in India.
yeah, I really love to be a researcher, but in the UK or anywhere else other than back home. If I'm going back home to become a researcher, I need a PhD. I need at least 10 years experience to even get myself heard.
Right.
We are not given the space to place our opinions. That's the first thing. And I don't know if I really feel like we're not being respected enough there.
Right.
Because I know I am just a bachelor student when I was back there, and I probably don't know much, yes. But again, I am a student. I'm
Right.
learning under you. All I expect is equal respect and the space to just talk what I feel,
Right.
like what I really think about. I need to be able to do that. So that's something I experience here. So with my master's professor, he's done so many preclinical trials. He's really good. I am a master student who's known about gene therapy for 4 months. That's it. But the first time I met him for this project, we sat down like this, and he asked me what I felt about his project, what I felt are the shortcomings, what could be done better,
Right.
and I did make some points, and he agreed.
Constant learner.
Yeah. I was given the space to
Right.
speak.
Right. Mutual respect. Constant
Yeah.
learner.
That's the thing. So all back home, we are not given the space to speak. I mean, some professors do. There are some people who are really good, but there are some people.
I
It's
get
just
it.
not
I
it.
get it. They just
And-
want to 295, go home. I know.
Exactly.
I get it.
So I really feel like back home, resources. True, we're not pouring this much money into research. That's true.
Is it changing? I think it's changing. a bit.
It could be changing now. Again. Hello. Hi.
Oh my
Get up
God.
again.
Oh, I can't feel my legs.
Oh, what the hell? There. So back home, we don't have. I feel like they're not putting that much money in. Probably, I don't know. Now they might,
Right.
but I left 2 years ago, and before that, being in Anna University, which used to do a lot of research, it was one of the front runners back in the south. I think it still is. So having come from there, they do a lot of research. And we even had like a separate lab away from the campus just to do dedicated research. But the amount of funding that my professors got back there will definitely be not as much as here. I can for sure say they're getting millions
Yeah.
over here.
Yeah.
And I am part of a project that has billions.
Right.
Because we are funded by the university, we are funded by 3 other organizations, there's more grants coming in. So they fund a lot here and they respect researchers. They want a lot from you. And the government also does respect it, I'm guessing.
Yeah.
Because they also fund a lot.
Yeah.
Back home, the amount of resources that we have is quite less. And the professors, they don't give you the space to raise your opinions unless you have 10 years experience.
Right.
Or you have a PhD.
Right.
You are a master's student, get out.
Right.
So I think it could change in the future. Hopefully, we change it.
Hopefully it
because
does. Yeah, that's what I'm looking forward to. As generations pass by, with more open-minded people,
Yeah.
don't go with the old culture and just great potential back in India, to be honest. So,
to be honest, like, I know most of my friends are from the same field. We are branching out, yes, but I know so many people from biotech background. If we go back, I used to say this, we go back one day, we start a startup, we have people for every department.
Right.
We will be the leaders. And people are good. Like,
Right.
Indians are really good when it comes to this. So I really hope that the next generation takes everything, goes back home, and gives it back when
Not the
the
next
time
generation,
comes.
us. Why not us?
Yeah, we can.
I am planning to. So, yeah. So, right. How? Yeah. Did we do a day in your life before? Yeah. Let's do that.
A day in my life. Okay. So a day in the life of a manufacturing technician producing an ATMP product is, I'll explain that, but that's not exactly what we are doing now. So a day in the life of a manufacturing technician who's producing gene therapy would be walk into the office, there'll be something called as a GMP envelope. So that is where you live. Okay? So you walk in there, no street clothes allowed. So get all your street clothes out. There's gowning that you do. So you gown up, and then there's rigorous sterilization, hand sanitization, all that goes on. You progress through different grades of clean areas. So as you move through different grades, there's either going to be more gowning or more hand sanitization.
Right.
And if you're taking material with you, that material has to be cleaned with different cleaning solutions at different grades for different time frames.
Right.
So by the time you enter the critical area, that is already like 20-25 minutes, and you would be in 3 layers of clothes, 3 layers of gloves, hairnet, mask, completely like an alien, and then you get in. So depending on the day, depending on the day of the process, what machines we interact with, it differs. So most of our processes are semi-automatic. So it is setting it up, click start, monitoring the parameters. So one of the most important jobs of a manufacturing technician is to be very vigilant. Anything could change at any point. Any parameter could go out of spec, and at the end of the day, it is still a machine.
Right.
It can do a mistake. So you have to be very vigilant. Sit there, record everything. We have like a BMR, a batch manufacturing record, that thick. Sit there, note down everything, and also to set up the equipment for the next day. That should also happen. And what else is done? Once you finish your process for the day, you have to clean the entire place. Not normally cleaning. Nope. We are trained in each and everything, so we are trained to clean. We're trained to clean.
Right.
That is something that I find difficult when I tell this to people. They're like, "You did a master's degree for cleaning," but it's a huge process, and I have spent 4 months of my life building a cleaning regime for this GTIMC with the least amount of experience, but it did work.
Okay.
So that's probably the biggest thing that I've learned this year. But yeah, my process that I've designed, but we also have to clean the place down with different cleaning solutions. Every time it differs. Every day it differs. Every week it could be different. Monthly there's a different schedule. And God forbid, if something goes wrong, if something comes out of our EM monitoring results that there's some kind of a microbial growth anywhere, it could be your gown, it could be a material, it could be somewhere. We scrub down the entire place from floor to ceiling with 5 different chemicals. The entire day goes in that. So it is really rigorous out there, and it justifies the name that it is a clean, it's called a clean room for some reason. So we do all that, that's our process, do all the process. There's a huge process that goes on. So I think our process is over a month, basically. It starts with upstream, where we grow the virus in little flasks first. And then from there, we grow the cells in little flasks. And then we move to a bioreactor, which basically grows it in a bigger scale, 50-liter bioreactor, that's what we have. And then we add plasmids, which is basically the viral components, let's say. And then that goes in, and then the cells are going to become viruses now, basically. They accept the plasmid, they form viruses. And that's the upstream. Now we've got the virus. Now we have to purify it. So that's the next biggest headache. You take that, you have to purify it like 100 times. We use something called TFF, tangential flow filtration. So we do that. Then we concentrate it. Now we have the virus, which is kind of pure. We've stripped off all the unwanted stuff. But the problem is there could be other stuff in it, and you're just introducing a plasmid. Some cells might take it, some cells might not take it. So if you inject a virus without the DNA, like the actual gene therapy in it, it could be harmful for a patient. The patient could even die. So we need to strip them off. So we go through another process called chromatography, which captures it clings to all the virus basically. There's a column, we pour the fluid, the virus sticks on. For other people, probably you might know.
10 standard chemicals?
Yeah.
Chemistry?
Exactly. So we strip the virus.
Nothing is going on waste, man. everything comes somewhere around.
Yeah, exactly. That's true. So we first get the virus out, separate the virus, and then from the virus, using different gradients of charge, ionic charge. We then take out the full viruses alone. And from there, we finally filter it to ensure there's no microorganisms. That's called sterile filtration. And then we vial it up. So all this will approximately take a month to happen. So with all this, there's still gowning, there's still cleaning, there's still environmental monitoring, there's still documentation. All this has to be done. And at the end of the day, even if you are in a 7-hour shift, if you go up, you're done with everything, you've cleaned your place, you've gone out, you're back in your street clothes, you still have to go up to the office and fill in some paperwork. There might be some kind of an event that happened, some kind of a deviation, you have to report it. There's a whole different way things happen in GMP, the biggest name. It's called Good Manufacturing Practices. So that changes everything. Even if you open a fridge for 2 minutes more than it's supposed to be open, that's a deviation. And you have a meeting for it.
Right. I'm not going to go and ask what you're working on right now, because I don't want anything confidential coming out of this. But I do want to ask, how was the application process? Interview? Do you remember any interview question? The application processes? If you want to say.
Yeah. So interview was application process. The University of Sheffield kind of keeps it simple. It's I think they give you one paragraph where you have to write why you want this job.
Yeah, supporting information. All right.
So just that. That's the interview process. Was just upload your CV. I'm not even sure if I uploaded my CV. It was just that one paragraph. That's all I remember. And basic questions, name and all that. Nothing else. And interview process was I do remember a few questions. It was mostly the same question that you asked me: how is a day in the life of a manufacturing technician?
Going back a bit, was there anything that the ATS? Was there anything?
I don't need it. So I don't need ATS certificate.
No, not that one. Not the security one.
Oh, okay.
I'm saying, while we apply, there's this machine that scans our resumes. Was there any strategy for getting through it?
So the thing is, resume wise, what I did was, so I did apply to a lot of companies initially. I started applying since June. And when I applied, basically, everything was rejection. So I was thinking what went wrong. And I changed my CV a lot of times. And what I did was, instead of writing paragraphs and paragraphs, I kept it simple. Undergrad, what are the skills that I got? I started noting down just the skills without being like writing a paragraph, writing a sentence, just bullet points. These are the skills that I had. Undergrad. Project, in this project, what are the skills that I had? So I started doing that. And probably after I did that, I actually got like 2-3 interviews. One was over the phone. Went really well. But they wanted 12 months industrial experience at least. So that went that way. But yeah, I think the thing that helped me was, instead of going too much, trying to sell myself, in the process of trying to sell myself, I started like writing a story over there. That's meant for a cover letter, not for the CV. So keep it simple probably. And then if you have whatever project you've done, ensure that point, these are my skills, bullet points, just that. That helps a lot. Now 1-page CV helps. A rambling one, they just go. I'm done. That's it. So mine was 1.5, and I tried to make it 1. Pulled out a lot of stuff. So I think that did help in terms of CV.
All right. Yeah. Then interview.
Interview was so I think there were like the entire manufacturing team at that point. They were there. The director was 6 people. I was interviewed for 40 minutes. Questions from 6 people, 40 minutes, all technical questions. I was almost on the verge of crying. But it was mostly a day in the manufacturing technician's life. What will you do? And since we still aren't manufacturing, we still are validating. So what do you expect to do now? That was another question. You're not going to manufacture.
What was these things? Were these things in the job description, or how did you research about this?
About the actual process. Okay, the process, it was in the job description, obviously. The job description does say you have to work in a clean room. And it also did say that we are not yet functional. So before that, you will be helping us with validation, setting up a GMP production room. But apart from that, the process wasn't there in the job description. So I think process-wise, I did my master's. So I had a little experience and learning about how things work and what could be the process. apart from that, I did my own research. This is how it is from the start to end. Nobody's going to say that. So I kind of made the process of completely from the beginning to the end, and I ran it by my master supervisor also, because he's in the same field, and he gave me some inputs, and that helped a lot, because they were actually impressed by that one bit that I said, because that covered everything. To be honest, I first was called for an interview in June or July for QC technician, not for manufacturing technician. And they asked me different
Quality
questions.
control in Gene?
Yeah, in GM, basically quality control in the same place.
Same place, all
So
right.
they will be testing whatever product that the manufacturing team produces. And at the end, if all the critical quality attributes are met, that's when they're like, yep, this is okay, it can go. So they do the testing.
What happened to that application?
So I did apply. I went there. They called me for an interview. That's the first interview that I attended my entire life. Very nervous. So at that point, I think there were only 2 people then, and everything went well. Answered most of the questions, but on the spot, they asked me if I would consider a manufacturing role, because they felt my skills will be more useful there.
Okay.
Because of my knowledge about the process. Because QC tech needed only the quality control attributes, but I was able to like give the entire process.
Life process.
Yeah. So that's it. I gave the interview. I came back. That's the only question they asked me at the end, and I got a rejection, which was 3 pages long. Very new. So I read through it. All it said was, "We loved you. We feel like your skills will be more suitable towards a manufacturing technician role, and we will be advertising for that very soon. So please do apply."
Okay.
And before that, we would also like to extend the opportunity for you to come to our place, see us working, and learn more about the process. That could help.
This happened to one of my friends recently as well. She applied for one job, got interview, but they had another opening for which they thought she might be more suitable. They offered the job.
Exactly.
I mean, that's really good. That's really good thing that they did.
I had to give the interview again.
Like,
It was annoying. But it was a different set of questions, but I kind of got the vibe from there. So this is what you're looking for. So you will know what the company is questioning after that.
what were the questions?
For manufacturing?
Yeah.
So
Process.
the process. First, it was the process, and after that, it was what you expect to do if not.
Right.
And then there were some technical questions like basic acronyms that we use in the industry. What is all this? Do you know about all this? And then events, deviation. Do you know? Because I'm a fresher. They kind of knew that, and they were trying to like understand how much I know.
Right. The core that you have.
Yeah. And they were like, what is a deviation? What's an event? And what do you do when this happens? What do you do when that happens? It was basically some scenario-based questions, some technical questions. The scenario-based questions were more towards you've not worked before. How will you handle the situation? Kind of a thing. And apart from that, anything else? It was, do you know how to write an SOP? Which is kind of the most basic thing that a manufacturing technician does. Like simple questions like that. But it was constant. And the director of our company,
Standard operating procedure.
yeah, SOP, standard operating procedure. So the director of our company, he basically asked me a few questions. It was, why do you choose this field? What is the reason behind it? Then what is, why do you want to work in this company specifically? What is your passion behind gene therapy? Things like all that, like assessing why you're passionate. That kind of questions are also involved. And at the end of the day, they also wanted to know if I'm going to consider a PhD at some point, if I'm going to leave them, basically. I think that was one of the most important factors for them, because I, coming from a very academic background, they were like, you have a really good profile. We don't want to lose you towards a PhD.
Right.
So that was one of the major concerns. So all that, we had a discussion basically. It was not even an interview, the second one. By that time, I'd gone to the place 3 times already. So they kind of knew me. So yeah, that was the second interview.
And they did sponsor you?
Yeah, they did sponsor me.
Right. That's good to know.
I got lucky.
Yeah. Yeah, it's a work you put in. It's not luck. So.
I mean, yeah, that's true. But I mean, if it was, I'm not sure if it would have happened if I got any other job. This job, with its funding, the amount of funding that's poured towards the GTIMC, that was a major contributing factor.
Yeah, true that.
Yeah.
True that. All right. How's the work-life balance?
It is actually really good. I am getting a lot of time. So in work, right now, so I think I did mention this before, like when you asked me what does my day look like. Right now, we're not manufacturing, we are just validating our facility. So there's a lot of things that I'm doing now. My role is a manufacturing technician, but right now, if I have to jot down everything that I've done, it's going to be so much different than what we expect. So we are validating everything. So there's validation of equipment going on. I'm doing documentation work, preparing SOPs, putting in like the basic workflow. We are a company that's started from scratch.
And it's a good place to be in. You're setting up the process.
Exactly.
That's what you need, actually.
I think that is what actually got me this job, because the first time I went to the QC tech interview, the head of QA was there, and he was the one who asked me, "We are a small company. Why do you even want to work here?" And the answer I gave was this, basically.
Right.
You are starting from scratch. So I want to be there to see everything. So that one day when I start my own thing, I would have been a part of the process. It's not like I have to hire people and ask them. Yeah, I know what to do. That's the whole point. And he didn't expect that answer. So that's what I'm doing now, and it's really good. So I am doing documentation, I'm going through validation stuff, I'm training with different kinds of equipment. At the same time, I'm also helping process development do their 2-liter scale process, which then we are also thinking about how to expand that 2-liter into 50 liters. So that's called scaling up. So we also have to think about that. We're also doing ordering furniture, ordering consumables. How do you do all this? The financing aspect of it, and setting up the normal workflow, like setting up a clean rooms cleaning regime, setting up how do you gown, how do you clean, all that we are doing it. So it's kind of I'm interfacing with the QA team, the process development, operations, and I'm doing manufacturing work also. So this is really good for me in terms of experience.
Great exposure. Great exposure, to be honest. Right. How are the people? How's the
people? They're really good. I love my team. So there is a manufacturing team, but we kind of consider the entire office as our team. It's just a small workforce. I think we're like 25, 30 people. We've just gotten to 25, 30. But everybody there is really passionate towards what they do. If you just start talking about something, the head of PD will hear it from there and he'll rush towards you. Like we were just talking about some equipment, we were racking our heads like, how do we do this? How do we scale it up to 50? And he was there. He literally like pushed his chair all the way. He came here, was like, I heard you talking about this. So my input. And then the head of projects comes in and he's like, oh, so you're talking about this. What do you want? I'll help you. So everybody's hands on. They're so passionate about the project that it's really good.
It's a really good environment.
It's a really good, like there's no toxicity at all. It's a really good environment. Even if you don't have any work, you just go sit there in the office and see everybody. That's coming for you. That's it. So it is a good environment. And because it's extremely small, everybody's putting in their effort. Like, be it you're the head of PD, I don't care. You still clean the lab because that's your lab. I'm a manufacturing technician. I'm not from the ops team, or I'm not a basic technician. But if something comes in, I carry it upstairs. I assemble it. That's fine. So when everybody is hands-on and passionate about the project, the environment is really good.
It's a typical startup environment.
Exactly. It's a typical startup environment. I am so blessed to have started my career here.
True.
So it's like, this is the first time I'm working, but I'm working in this environment. Is it supposed to be this good? Okay, probably it is. But yeah, that's the thing.
And the flexibility.
Oh, that like I'm so if I can work from home anytime. We have, because I'm a university staff still, we have like specific holidays per year, these many numbers, all that's there. But still, if today I decide I go into work, I'm sick, you can leave. It's fine. You can take sick leave, you can work from home, but if you're sick, please don't work from home. I got grilled by the head of QA. The head of QA kind of likes me a lot. He kind of thinks of me like, he's my project kind of thing. So he, I think the first few weeks, I did a lot of work for him. And he saw me answer an email at 9 o'clock. I think it was a Friday or a Saturday.
Don't do that.
9 o'clock. It was not even answering. It was basically accepting his meeting on Monday. In Google Meeting. That's all I did. And I think on Tuesday he called me and he was like, so this meeting, I saw you have accepted it at 9 o'clock on Sunday. Why? I said, I was just up. I always have my phone.
Right.
I live alone. I always have my
Right.
phone with me. So I was just looking at it and I just accepted. Nobody is supposed to do work beyond 5 o'clock. Saturday, Sunday, you are not allowed to do work. He practically like schooled me there. He's like, even if the head of the GTIMC walks up to you and says you have to work, I need you to do this on a Saturday, you say no.
Yeah.
So they are like really good people. They
Kind
just
of
don't
the
care.
same thing happened on my first day in work. Literally, they said, "Man, they'll kick me out."
Yeah.
So yeah, that's that. Any final thoughts, tips that you want to give the listeners out there?
Okay, so this might be for science graduates, but
Yeah,
anybody can
specifically your
use it.
field or anything.
Yeah, so science graduates, basically there are two fields, academia and industry.
Yeah.
It's kind of, there's always a debate about this.
Right. Academia, go to University of Sheffield. Industry, I don't know. I'm just in kind of a split decision here. For probably gene therapy and stuff, still go to University of Sheffield. I don't know, maybe computer science and stuff. No. Mechanical engineering, yes, we have good research facilities here. Right, yeah, go on. Sorry.
That's right. So, especially for science graduates, so there's academia, there's industry, there's two different parts that they see. The thing is, people mostly say, you're going to the industry, you're selling your soul. Please don't go there. That's kind of thing that academics
Right.
always believe in, and they say that. But I'm not going to say that. All I'm going to say is, you don't have to choose what path you're going in. Like, it is just the next step. Your goal could be, I want to be a supervisor, I want to be a PI, I want to be a principal investigator fighting a gene therapy, be it. Or it could be, I want to be a CEO of a company. Whatever it is, right now, you have to just think about what's next. And that next could be in academia, it could be in industry.
Right.
You can start up as a research technician. You can start up as a manufacturing technician. You can always switch up. I can work 3 years here in the industry, go to a PhD, do a postdoc, come back to the industry. Or you can just stay there and go in academia. So you can always switch up. You don't have to be like, oh my God, this is the most important decision. Am I going to academia? I'm going to industry. I have to decide. That's not it. You can always switch up. That's something I learned this year. So
it's kind of same to one of the stories that I heard. Something like a horse and a boy is walking through the desert or the forest, I don't remember, during the night. And the boy goes to the horse that, I can't see ahead. I can't see anything ahead. And the horse comes back saying, can you see the next step that you have to take? And he said, yes, then take that.
exactly. Just focus on the next step. And this is not like, if you enter academia, it doesn't mean that you're never going to the industry. That you can always switch up. That's one thing. And the other thing is, a lot of people have asked me this because I have gotten a job for what I basically studied. A lot of students at the university, they kind of ask me this. Most of them are freshers. They're coming here. Will it be good to have experience? For my field, what I've heard from recruiters, what I've personally experienced is 12 months experience in the industry, amazing, really good. If you have that, if you can have that, well and good. If not, if you're still a fresher, don't have to panic. You don't have to lose your mind. At the end of the day, you still have the skills that you have. So if you're going to an interview, if you're applying to the job, as I said, in the CV, it's just your skills that matter. Ensure that any project that you've done, list out what are the skills that are associated with the projects. And in your cover letter, all the recruiter is looking for is how you say these skills could be transferred to this job that I'm doing. Explain that in your cover letter. This is the skill, I can transfer it this way for this job I'm doing. That's it. So that's step one done. You're explaining to them why you can do this. So focus on your skills and think about how that skill will help you do this job. Express that. That's enough. So that, and when you're giving an interview, it's all about your confidence. You do have the skills. So just confidently speak, yes, this is all I have and this is what I'm giving you.
What I can do.
Exactly. And if you have that confidence, if you're expressing your skills and if you're clearly telling them, this is what I have, I'm transferring this to your job in this way and this is how I can be of use to you. And that's it. If they ask any questions that you don't know, I do not know this. This is all I know and this could probably help me this way. That's all you have to say. And even if you don't get that job, you still would have made a huge impact on them. That is all that matters. Your confidence would have blown them away and they're like, oh, okay. And in science field, mostly connections and networking helps you a lot. So even if it's a rejection, make sure you leave your mark there. They do remember you.
Right.
That will somehow help you. That's it. They'll even offer you a job later on, but it will always help you. So even if you're freshers, please don't panic. That's the only thing. You are yourself. You have your skills. Just sell that. Be confident, and that's it.
Right.
You'll flow.
All right. Thanks for doing this. Thanks for taking out the time. Thanks for not doing it tomorrow. This is a weekend. That's it.
Thank you for not doing it tomorrow.
Sure.
Thank you so much for having me.
If you like the video, please subscribe.